Varad Vijayakumar, who is Associate Director Manufacturing and Aseptic Fill Finish at Terumo Medical Care Solutions. And with that, Varad, the virtual stage is yours.
Hi, uh, thanks Igor for the nice introduction. I will share my screen quickly and jump into the presentation directly. Okay,
Could you please confirm that the sound is okay? Thank you so much.
can you able to see my screen?
Yes, perfect. You can proceed.
Okay, one moment. Yeah, okay, let's start now directly. So today's topic, good morning, good afternoon. Welcome to this webinar on aseptic behavior in isolator-based filling lines. So today we will focus on the decisions people make when direct supervision is absent. Okay, so I will also point out my uh, laser one moment. Yeah, it works now. So in sterile manufacturing, those unseen moments often define the real strength of the contamination control system. So this session is about behavior, culture, and patient safety, not just procedures and compliance. So disclaimer, so this webinar is intended purely for educational and professional development purpose. The examples and references used here are based on publicly available regulatory guidance and industry case materials. No confidential property or company specific information is being shared. Please treat this session as a learning resource, not as a formal legal or regulatory advice. Okay, then we move on now. About myself, my background includes aseptic processing, contamination controls, media fields, isolated technology, and regulatory compliance. Over the years, I have worked across multiple companies and international environments in sterile manufacturing. My focus has always been the same: how systems behave, how people behave, and why failures happens quietly. That practical experience is the foundation of what I will share today. So I also have a book written by me. You can also use this QR code to connect with me in LinkedIn so that all the details are, if unleft questions can be answered. Okay, welcome for this webinar actually. So this session is mainly for the operators, QA colleagues, QC supervisors, and engineers, validation teams, whoever it is. Aseptic behavior is never owned by one department alone. Sterility assurance is a shared responsibility, guys, across the entire organization. What each person does in routine moments has a direct effect on the patient safety and product quality. We have to remember this always. Each person does in routine moments has a direct effect on patient safety and product quality. So today's agenda and learning objectives, what we will gain from this webinar. So we will begin with the foundation knowledge, technical procedures, and isolator practices. From there on, we will explore behavioral psychology, regulatory expectations, and practical decision making. The aim is not only to explain what the rules are, because we all know that, but also why they matter. That's important. By the end of this session, you should be able to make better aseptic decisions even when nobody is watching. That is our theme for this today's webinar. By the end of the session, you will understand not just what, how aseptic behavior more, but more importantly, why enabling you to make sound decisions even when nobody is watching. That's our main theme for this today's webinar. So this is the quote we need to read always. A clean room alone does not ensure sterility. Product safety depends on qualified design, validated controls, and disciplined personal practices. Okay, very important. We invest in isolators, HEPA filters, and decontamination system, but behavior still remains central theme. Technology supports sterility assurance, but human discipline is always critical factor. Let's move on to the next slide quickly. What is aseptic processing? Aseptic processing means manufacturing a sterile product without final terminal sterilization steps. That means sterility must be protected continuously throughout the process. There is no final kill step to rescue an error made during filling or assembly. We need to keep remember this. Every operator action therefore becomes either a contamination risk or a contamination control point. So please keep this point in your mind. Aseptic processing is manufacturing of sterile drug products without final terminal sterilization step. Okay. Okay, now the important slide comes. The right mindset for the aseptic processing area. So this slide reminds us that the mindset required in aseptic manufacturing is different from the normal behavior. A moment, it's not working on one. Okay. In daily life, people move quickly and take small shortcuts. But in a aseptic area, those habits become contamination risk. The moment you enter the controlled environment, your behavior must become more deliberate, disciplined, and self-awareness. Okay, so please keep this in mind. Daily life mindset is not the same as aseptic area mindset. All these things what we do is in our daily life, but what's happening in the aseptic processing area, we need to follow a proper disciplined behavior. Okay, sorry, it was so quick. The rules and behaviors change when you arrive to the aseptic area. So this actually we do in daily life, but when it comes to clean room, you have to behave properly. Okay. Okay, from this slide, what is wrong here? What can impact the product and patients? What is the good behavior? I'm not going to ask any questions to you all, but you just need to know what's wrong here. You know the answer better. What person is doing here? What person is doing here? What the person is doing in this room with the goggles, and what's wrong here. Maybe some people may not identify here, but it's about the improper gowning. Some of the goggles or the skins are exposed here. So you know now what is important issues from the aseptic behavior point of view. If you see our next slide, um, now the answer is people are the largest or highest risk factor and the source of microorganism. Okay, people remain the biggest contamination risk in any sterile manufacturing system. Movements disturb airflow. Poor techniques blocks the first air. What we will see what is first air in next slide. And we are encouraged to some shortcuts. Overconfidence. This is very important. Very important. I always speak with this point in any of my webinars. So overconfidence. Experience does not eliminate risk. Believing I have done this hundreds of times. Okay, leads to carelessness and failure to follow proper procedure. Some people always say that I have done this hundreds of times. That's not like that. A strong aseptic culture recognizes that human behavior must be managed as carefully as equipment environment. So human behavior is very important, and it is humans are the highest risk factor in our clean rooms or in the aseptic areas. So today we are talking mainly about the isolator based filling lines, which is grade A and grade C background. We are not talking about the conventional clean rooms today. We are concentrating on isolated, but all this concept remains for both the traditional and isolator based filling lines. Okay, now we will see what is airflow and first air concept. First air is the cleanest air available because it comes directly from the HEPA filter to the critical surface. Our responsibility is to protect the path and avoid blocking it with the hands, tools, or materials. Whenever operator do any interventions, any activities in the critical zone, please keep in mind that you try to avoid the first air blockage. Okay. Whenever first air is interrupted, contamination risk increases immediately. Okay. Good aseptic technique always means working with airflow, never against it. So you can see here, you have to follow, you can follow this, but do not block the first air from your hands, glove ports, or something like that. So it's very simple definition. What is first air? First air is the cleanest air available because it comes directly from the HEPA filter to the critical surface. Our responsibility is to protect that particular path. Okay. Okay, next slide. I'm just giving a background now, slowly as a foundation, and later on we will move more deeper into psychological part, what happens when nobody is watching. Okay, why aseptic behavior matters in pharmaceutical manufacturing. Aseptic behavior is not only a compliance expectation, forget it. It's a direct patient safety obligation, guys. Injectable product bypass the body's natural defense system, so contamination can have a severe clinical consequences. One contamination event can affect an entire batch and potentially thousands of patients it will get affect. That is why even a brief lapse in discipline can have a consequences far beyond the filling line. So we always need to see a single contamination event can affect hundreds of lives. Okay, so please keep this in mind. If contamination occurs due to a momentary lapse in aseptic technique, consequences cascade through the entire batch. Single contamination can affect entire batch, affecting potentially thousands of patients across multiple countries. Okay, and also finally, sanctions, facility shutdowns, irreparable damage to the public trust in pharmaceutical manufacturers. Finally, business is lost. So please remember, small things in the aseptic area matters a lot. Okay, next slide, we will go to a global regulatory landscape. I just referred some of the good guidelines here for your own reading. And this is also one of my upcoming book for the manufacturers there, where they can able to see lot of observations, the response, poor versus strong, etc. It's very good book coming up, but it takes some time. But if you see here, regulatory landscape, all major regulatory authorities place a human behavior at the center of sterile manufacturing control. FDA, EU, MHRA, WHO, whatever it is, the contamination, the message is very clear. And message is one: contamination control must address people, process, and system together. Good aseptic behavior is therefore not an optional, guys. It's a global regulatory expectation. It's not an optional. It's a global regulatory expectation. Okay, please remember this. All these guidelines says one clear message: contamination control must address people, process, and system together. Let's move on now. We have a lot of slides, but we'll try to complete within the timeline. What EU GMP Annex 1 2022 changed and what it says. Okay. Um, Annex 1 2022 revision is very big revision, which is like 59 pages currently. And it requires a formal contamination control strategy that explicitly includes human behavior as a contamination risk. It also ensures that isolator usage, realistic media fails, PubSit requirement. We will talk more about later on, not in this session. Or what about PubSit requirement, mandatory requirements? The key message is that modern compliance is no longer just about following the steps. That's important. The key message is that modern compliance is no longer just following the steps. It is about understanding and controlling the risk. That is what the EU Annex 1 says. Just not follow the steps, but you need to understand why and what are the risks involved. Okay, now I will give you some background about grade A, grade C environment. That's what we are going to talk today. This slide is very clear that grade A is the sterile core where direct product exposures takes place under highest level of environmental control. Okay, these are some important points I have mentioned here for the grade A sterile core. What is grade C background? So grade C supports the surrounding operations and helps protect the primary sterile boundaries, which means this protects the sterile core area. These areas are interconnected, not independent. Okay. If either side is poorly controlled, if either one is poorly controlled, the overall contamination control strategy becomes weaker. You have to control both in a proper way. If this is perfect, this is not perfect. If this is perfect, this is not perfect, then your contamination control strategy becomes weaker. I will move on to next slide. What happens when protocols are compromised? So these written procedures are compromised. What happens? This slide shows how a small deviation can grow into a major product quality event. A small deviation leads to a major product quality event. It usually begins with a shortcut. Whenever we do some shortcuts or something we are ignoring the requirement, followed by the contamination, introduction, and product exposure, then what happens? That can lead to patient harm. Investigation and recall happens. In aseptic manufacturing, minor deviations have a potential impact, guys. Small, small event, big impact. Then investigation and recall happens. So it will take a lot of time to close this event, close these issues, and to become normal, it takes a lot of time. We have to recall the products. So please be careful whenever small things are happening. Please pay attention to your behaviors in the aseptic clean rooms. Now I will touch a little bit about the isolator. That's what we are going to talk about. So what is isolator? In a simple words, I will not read the entire slide and make you bore, but I will give you a simple definition. Okay. An isolator is a physical barrier system that separates the product and critical zone from the surrounding room and the operators. That's it. It maintains a grade A condition using HEPA filtration, pressure control, and validated biodecontamination or VHP cycles. Operators interact through glove ports instead of direct open access. Okay. This reduces a contamination risk significantly, but it does not eliminate the importance of correct human behavior, guys. Very important. You have isolator, you have wraps, gloveless, everything, but behavior is important. Okay. Because operators interact through glove ports instead of direct open access. But still, the contamination is still there. It depends totally on your correct human behavior. Okay. There is a simple comparison between wraps and isolators. Okay. Wraps and isolator both reduce the contamination risk, but they do at different levels of separation and control. Okay, we have seen what is isolator. I will not talk much about wraps. Wraps still depends more heavily on room conditions and operator discipline during intervention. Isolators provides a strong separation and with validated decontamination capability. However, guys, neither system can compensate for poor technique or weak aseptic discipline. You have both, but very important is aseptic technique. It cannot compensate. Okay. This will support us, but our behavior is important. So grade A environment, the sterile core. Now we are in grade A environment. Then we will talk about grade C environment quickly. These are something you can read it by yourself, but I will just simply say grade A environment is the highest classified environment used in an aseptic filling operation. It is defined by strict particulate limits, unidirectional airflow, and constant environmental monitoring. This is the zone where product, container, and closure are most vulnerable. Okay. Any operator action in this area must therefore be intentional, controlled, and scientifically justified, which means with your risk assessment or whatever you do, whatever interventions you are doing inside the isolator in a grade A environment, it needs to be controlled and scientifically justified. You cannot simply put your hand inside the isolator and do some interventions during your product manufacturing. Everything needs to be scientifically justified. Why are you doing that? Okay. Rest of the informations are very good for good reading purpose, but I will not read entirely. I'll just say what is the core message. Next, grade C background supporting clean rooms. Grade C areas support the isolator and clean room operations without directly serving as the sterile core, because this is just a background environment. It will support, it will be like a protection layer for grade A environment. They are used for staging, preparation, and controlled personal and material movement. Although the less critical than grade A environment, they still play an essential role in contamination control. Okay, if grade C performance degrades, it increases pressure on barrier system and the overall process. So it's very important, guys. So if the behavior or the grade C performance degrades, it will impact grade A environment also. So that's what I mentioned in this green highlight here. The relationship between grade C background and grade A core creates an integrated contamination control strategy where neither zone can succeed independently. Okay. Compromise grade C condition increase contamination pressure on grade A barriers. That's important. Grade A intervention that violates isolator integrity expose sterile zones to grade C environment. Understanding this interdependence is crucial for maintaining aseptic behavior across all manufacturing zones. Please remember this. This is less critical, but it is very important for supporting grade A environment. Okay. Okay. Next slide. We know HEPA filtration system and their role. 99.97%, zero point 0.3 critical size, 24 by 7 operations in the clean rooms. So I also mentioned integrity test material for people who wants to know more, but I will not read that. I will touch little bit. HEPA filtration is one of the main technical foundation for aseptic manufacturing. These filters remove 99.97% of particles at zero point 0.3 microns, the most penetrating particle size. Okay, they work through interception, impaction, and diffusion. These are some methods how they work depending on particle size and airflow behavior. Operator responsibility, if you see here, operators may not maintain the filters directly, but they help protect them through proper use and awareness. Okay, so they have to take care, as we discussed before, for first air and airflow concept. So operator responsibilities, while the HEPA filters maintenance falls to engineering and validation teams, operator play crucial roles in protecting these systems. Avoiding contact with the filter face, reporting unusual odors or visible damage, monitoring pressure differential trends, ensures pre-filters are changed on schedule, all contribute to HEPA long lives. Remember, damaged HEPA filters cannot be repaired, only replaced. There are some articles where we can be able to patch the HEPA filters, but only replacement is expected at significant cost and production downtime. So everything is important. So operator responsibility, if something goes wrong, you need to report it immediately in terms of HEPA filters because it's very critical, as I described before. Okay, now we also know we have discussed about isolator, we are talking about the biodecontamination cycle, etc. But what is exactly this slide tells us? Before production, isolator must undergo validated VHP cycle or validated decontamination cycle using hydrogen peroxides, okay, vaporized hydrogen peroxides. The cycle typically includes preconditioning, dehumidification, dwell time, aeration. Its purpose, what is the purpose of this cycle? Its purpose is to achieve a validated 6-log reduction of resistant biological indicators. That's important. Okay, 6-log reduction. We will don't ask me what is 6-log reduction because that also has a separate discussion. A detailed understanding is required. But in general, the expectation from VHP cycle is 6-log reduction. If the cycle fails or it is incomplete, the system cannot be considered ready for aseptic operations, right? It's very important from this slide. That's what I'm trying to say. Operator responsibility, please know why. Okay, not just a VHP cycle needs to be done. It needs also to reach 6-log reduction. Okay, personal training requirement and competency assessment. Aseptic training is not a one-time event. Okay, it is a structured development process. Operators move from foundational knowledge, basics, supervised observation phase, with shadowing experienced operators, observing proper techniques, asking questions, understanding the workflow. This is just my understanding, okay? No, don't get anything from here, but it's just my experience. So first 2 to 4 weeks, operators should know all the basics. Then they are under observation phase and they are under supervision phase, like performing operations under direct supervision, receiving immediate feedback, developing muscle memory, what's going on for the operators. Then assessment, written examination, practical demonstration, media field qualification, demonstrating entire competency for 1 to 2 weeks. Then it's a continuous development. This is the phase where we are missing sometimes. like annual requalification, refresher training, techniques and refinement, emerging technology and education. So for all this, you need to spend some time for the continuous development. Okay. Media fields remain the concept, the strongest practical test, but not only media field will support always, but you need to have a proper behavior, aseptic performance, core competency areas, important points I have mentioned here. Media field, about media field, I have given some information here. But overall, foundation phase, observation phase, supervision phase, finally assessment, and then continuous development. Now we are slowly entering into our psychology and unsupervised behavior. That is our theme today. So people behave differently when they believe they are not being observed. Very important, guys. There is a behavior change. You can see here 20 to 40% behavior change when individuals believe they are being observed. And people change their behaviors when they know that nobody is watching them. In aseptic manufacturing, that creates a serious vulnerability because contamination is often invisible. We will talk about that later on, and delayed effect. Okay, contamination is often invisible and delayed in effect. This makes it easier for people to rationalize some small shortcuts or think that nothing happened last time. I have done this for a long time. That is why culture, accountability, and intrinsic discipline are so important in sterile operations. Okay, people don't see something. We will talk about that also in our next slide. So, very important point. What I'm trying to say here: behavior change. 20 to 40% of behavior change when individuals believe they are being observed. If operators are not being watched, I'm not saying they will perform every time 100%, but there is a slight behavior change. This is as per the research finding. 3 to 5x deviation likelihood increases under time pressure without supervision. Very important. 3 to 5x times the deviation chances are increased. Okay. That's the main point from this slide. Okay, common shortcuts and their devastating consequences. Most contamination events can be traced back to moments when convenience was chosen over discipline. Okay, inadequate hands hygiene before intervention. There is a shortcuts. Operator skips sometimes hand sanitization before inserting hands into glove ports. Okay, reality. Glove ports integrity depends on glove. I will read one. Okay, glove port integrity depends on glove condition, proper technique. Micro perforations, insufficient glove overlap, glove tear during operation can transfer hand flora directly into grade A. Okay. Study shows hand harbor 10 to the power of 4 or 10 to the power of 6 CFU even after hand washing, sufficient to contaminate entire batches. So please sanitize your hands before you perform your intervention. Surface decontamination shortcuts, improper cleaning the surfaces, bypassing material transfer protocols because sometimes external sanitization for the materials are not performed properly. Skipping the gowning procedures properly, like improper gowning. We have seen some images before. So those are all the shortcuts happening and lead to a bigger consequences. Individual shortcuts compound rather than simply adding risk. An operator who skip hand hygiene or the surface cleaning or the glove or the particle alerts triple contamination risk. They create risk where each shortcut amplifies the consequences of others. This is why minor deviation can't be tolerated in aseptic manufacturing. They create cascading failure modes that overwhelm contamination control. So what I'm trying to say here is do not do any shortcuts. This is just some examples I have mentioned here. In aseptic manufacturing, repeated small compromises can create a very large contamination risk. That is the key message you need to take, okay, from this slide. I tried to make one nice case study. You can I will spend some time on this slide. Okay, this case study shows that contamination events are often caused by ordinary people under pressure, not by intentionally careless people. Okay, so let us go through this case study quickly. Thursday evening, 10 o'clock, late shift, for example, senior operator Varad, my name, arrives for night shift. Okay, And he saw that everything is running well, everything is going fine. He takes pride in his work, wants to help team meet the commitments. Okay, he's also a well experienced, 8 years of experience, well trained. Okay, what happens at 23:15, first decision point. During material transfer, Varad notices the RTP decontamination cycle failed to start properly. Standard procedure requires aborting transfer and call for engineering. However, engineering is not available and calling them means 2 to 3 hours of delay. He thinks that, okay, it's a small transfer, okay, 2 to 3 hours of delay will happen. Then Varad thinks that I have transferred materials hundreds of times in past. I will be extra careful with surface spraying. I can only use surface spraying with sporicidal or anything with IPA, something like that. This one time won't be a matter. He proceeds with a manual transfer without decontamination cycle, thoroughly spraying all surfaces, but he has sprayed everything. Okay, this is the first issue. Then second issue, when the filling operations proceed normally until particle counter shows elevated reading near filling needles. There was an excursion in the particle counter. Varad knows he should stop and investigate, but filling is going well and only 2000 vials are left. He thinks probably just a sensor drift, okay? Overconfident now. We will investigate after the batch. He continues the filling, documenting the elevated readings, but not investigating the root cause. He did not stop the line. Then what happens? Now 500 vials are remaining. The critical moment comes. Whereas glove tears during the stopper placement, there is a glove damage. Protocol requires stopping immediately, changing the gloves through validated procedures. Okay, or do not use the gloves. Now he has exhausted. Okay, he's under pressure. Now he thinks glove change will take 45 minutes, including the decontamination. And then he says, okay, my hands were sanitized. The tear is very small, pinhole, for example, or something like that, and away from the fingers. I will just be extra careful for these last vials, 500 vials are remaining, and he completes the filling with the damaged glove. Then what happens? Now the consequence actually shows. After 2 weeks, there are 14 contaminated vials during sterility testing, a failure rate of 0.7 percentage, well above the 0.1 percentage is the limit. Okay, less than 0.1, which means zero. The investigation begins. Environmental monitoring data shows the particle count trend from here is excursed. Varad batch records are reviewed. Under questioning, he admits the protocol deviation. He admits that there are some deviations happened. The entire 20,000 vial batch is destroyed. Now what happens? He faces disciplinary action despite him previously excellent record. Okay, so small decisions. He's a good operator, he's a very good person in terms of aseptic techniques or everything, but a small action, small decisions made him as a faces a disciplinary action. So here what I'm trying to say is not a bad operator. He's experienced, trained, committed to quality, but he faced a series of decision points, 1, 2, 3. Their production pressure, fatigue, insufficient support created conditions where shortcuts seems reasonable. Because of improper support, pressure, production pressure, tiredness, all this seems to lead to a shortcuts. Each decision builds on previous one, creating a psychological commitment to complete the batch despite having mounting evidence that conditions weren't right. Okay, very important. This one time won't matter. I will be extra careful. Probably just a sensor drift. These are internal dialogues that most of the lead to most of the contamination events. They seem reasonable in the moment, but ignore the fundamental statistical nature of contamination. Any deviation creates risk, multiple deviation compound that risk exponentially. So what is the lesson here? This case illustrates the contamination events rarely results from incompetence. They emerge from normal human psychology operating in the system with inadequate safeguards. Preventing these events requires a designing system that make right things easy and the wrong things hard, creating culture where asking for help is strength rather than weakness, and eliminating goal conflicts between production and quality. Please follow this. Production pressure, insufficient support, normalized deviance, fatigue effects, supervision gaps, culture issues. So overall, the case study says that it's because of many factors. Please be very careful when you are taking decisions in the clean room. I hope you would have enjoyed that case study. Okay, so what visible and invisible contamination risk? Okay, one of the hardest parts of aseptic work is that the most dangerous contamination risks are invisible. So what operator sees, what operator don't see. They cannot see microorganism, but operators can see broken parts, missing components, or obvious physical damage. But what they don't see, microorganisms, subvisible particles, surface bioburden, airborne contamination, glove port perforations like pinholes. They can't see by their naked eyes. Endotoxin contamination. So these are all invisible, but operators can able to see all these things. Okay, that is why aseptic work must rely on principles, monitoring, and discipline rather than visual confidence alone. Okay, this is a very good thing. We can read it later on. There is a scenario here, but the core concept is whatever you see, and there are more critical things which are not seen. Okay, now let's move on to cascade effect of single contamination events. A single contamination event can trigger consequence far beyond the immediate batch. It can lead to investigation, product impact, shutdowns, kappa programs, regulatory scrutiny, and significant business loss. A single contamination event can lead to business loss. 50 million average total cost per contamination event includes all direct and indirect impacts. 2 to 5 years to recover. Okay. Typical type. There are a lot of companies still cannot be able to recover from the poor aseptic techniques. 1,000 people are affected. There are a lot of cases involved in globally that there are a lot of people got affected because of contamination events, single contamination events. Okay. Please keep this in mind. This will product impact, then you will investigate, then production impact, you cannot produce anything. There will be a lot of corrective actions, regulatory response, your name is getting spoiled. I mean, you means the company name, and finally the business is lost. Now let's move quickly on later some of the important topics related to operators. Proper not only operators, for all the clean room persons. Proper gowning procedure for isolator operations. Gowning is the first personal contamination control barrier between human body and the clean environment. Okay, gowning is very important. That is the primary contamination control barrier. Correct gowning includes hygiene, proper sequencing, glove application, and final inspection before entry. Each layer has a purpose. Okay, each gown has a purpose. Skipping any step weakens the whole barrier concept, guys. Do not skip anything. Good gowning should be treated as a disciplined aseptic process, not as a routine dress code activity. Okay, there are some common gowning failures: incomplete hand hygiene, hair exposure, garment reuse, exposed skins, torn garments, skipped steps, touch contamination. We have seen one good image in my previous slides. So do not skip anything. Small actions are very important. That's what I'm trying to say from an important slide. Proper gowning procedure, which means this is the primary contamination control barrier between human body and the clean environment. Okay, now let's move on to next slide. Very interesting one. A simple concept again: hand hygiene protocols. Hand hygiene protocols before and after interventions. So we all know hand hygiene is one of the simplest and most powerful tools for contamination control. Hands carry both a transient and resident microbial flora, which can transfer to gloves and then to critical surface. Okay. Effective hygiene requires a proper washing, IPA sanitization, repeated application when needed. You have to repeatedly do your hand sanitization, and correct glove change practice. Okay, healthy skin condition also matters definitely, because damaged skin increases contamination risk. So always follow a sequence of steps properly as per your procedures: wet, scrub, soaping, and rinse and dry completely before you enter into the clean rooms. Okay. Very good slide for me, very important one, and for everyone. Correct use of glove ports and sleeves. So we all know that this is just a funny image. Don't take it in other way. Glove ports are the main human interface with the grade A isolator environment. Okay, their integrity depends on correct inspection, insertion, positioning, movement. Okay, and withdrawal techniques. Very important, very important. Integrity depends on correct inspection. Please take this word very clearly. Proper insertion of the glove ports into the isolator, proper positioning, proper movement, and withdrawal techniques. Operator must avoid stressing the glove materials, okay, overreaching or contacting the sharp surfaces. Any doubt about the glove integrity must be triggered immediate action because continuing with a damaged glove is unacceptable. When you find something wrong in your gloves, please stop the action. Inform to your supervisors. Okay, you have to change or as per your procedures. Okay, so that's what I'm trying to say here is hand insertion, positioning movement, tool and equipment handling properly monitor your gloves, and finally when you take out, it's very important. Okay, glove change protocol. Any visible damage to glove ports requires immediate glove change following validated procedures. Glove changes typically involve stopping operations, decontaminate. These are some additional information which I provided people who do online changeover of the gloves. Okay, don't think about production has stopped. Okay, we have to see mainly about the contamination control. If it delays for 60 to 90 minutes, it's okay. Our main objective is patient safety. Okay. Next, I also want to touch a little bit about different primary packaging material handling. Handling of vials, stoppers, and creams. Primary packaging components must be handled in a way that preserves sterility and closure integrity. Whenever you handle your vials or whenever you handle your stoppers, you have to always ensure that our first objective is sterility. Okay, vials should be handled by the body only. Okay, I mean if you want to catch the vial, you have to only catch the body of the vial. Okay, stoppers by the outer rim. When you take the stoppers, you have to take from the outer rim of the stoppers. Okay, and crimps by non-critical external surfaces. Critical surfaces must never contact with the cloud hands or some non-sterile tools or some non-sterile forceps. Okay, so please be careful when you're touching your critical surfaces. So as I said before, while interior and sealing flange surfaces, topper sealing surfaces, crimp seal interior interior contact should not be touched. Never. I also mentioned some important process how the vials are handled. It gets sterilized. It comes out from the tunnel. When handling the individual vials, operator must grab the vial by the body, never the neck or flange. Maintain awareness of vial orientation. Okay, always you have to ensure before you touch that particular product, primary packing material. Avoid any contact with the filling needles or the other equipment that might compromise the interior sterility. Dropped vial must be discarded regardless of whether breakage occurred. Okay, don't use the dropped vials. Then I moved slowly to stopper handling. Rubber stopper elastomers, as I said before, how to touch that. Crimp application, you have to touch the external surface. That's the point from this slide. Okay, then I'll quickly go to the aseptic manipulation technique inside the isolator. Aseptic manipulation is one of the highest risk activities because it places the operator directly into the critical process interface. Okay, there are a lot of steps I have mentioned here. First principle, second, third, fourth. The basic principles are to minimize the interventions before minimize the interventions, and second is like plan before acting. Third is move smoothly. Okay, and maintain the airflow awareness. I have mentioned very detailed procedure here, how to do all these phases, principles one by one. Every manipulation should be necessary, controlled, and documented when appropriate. The best intervention is often the one that was prevented through planning and system design. Okay, whenever we do any interventions, we have to be properly planned interventions. Okay, tool selection, visual verification, duration awareness, documentation, everything you have to plan properly and do your interventions in the grade A inside the isolators. There are some many important points about the video recording, depends on the country. but that's all for the uh, as per their country requirements. Common manipulation scenarios also I have mentioned here: sampling activities, equipment adjustment, jam clearance, everything. How you have to stop the activity. That is the main concept here, and ensure that nothing has been left in the track, and do your intervention in a proper way with a proper tool selection, visual verification, and awareness when you perform the intervention. Environmental monitoring and alert responses. Environmental monitoring gives real-time feedback on whether the aseptic environment remains under control. Yeah, we all know that. It includes particle counters, pressure differentials, airflow checks, and temperature humidity monitoring, guys. Okay, we all have to follow this, and we also need to trend it properly. The value of the system depends on how people respond to alarms and trends. Very important point. Value of the system depends on how people respond to alarms and trends. An alert is not a background noise. It's a signal that system may be drifting towards failure. If you get any alarm at the background, if you ignore it, then there is a big consequence. Whenever there is an alert at the background noise is coming, it is a signal that the system may be drifting towards failure. We need to be very careful and respond to those alarms in the clean rooms. Okay. Alert detection, immediate response, then you have to investigate, then you have to include the corrective action, and then follow up properly. Okay, that's very important in our clean rooms. Documentation requirements and during operation, we all know that batch production records, environmental monitoring logs, deviation reports ensure that documentation is a formal evidence that operations were performed correctly, and we all know this alcohol principle, etc., etc. The very important point I'm trying to say: batch records, environmental logs, and deviation reports must be completed accurately and in real time. Okay, because backfilling from memory creates data integrity risk and weakens the quality system. Backfilling, do not backfill anything. In sterile manufacturing, good documentation is not an administrative work, guys. It's a part of sterility assurance. Whatever you document, it is just not an administrative work. It's actually a part of our sterility assurance, ongoing sterility assurance. Okay, whatever we have done previously, now whatever I explained in my previous slides, now it comes to documentation requirement. You have to record it in a real time. We all know this data integrity, alcohol principle, etc. But the core concept, what I'm trying to say here is it's not an administrative work. It's a part of sterility assurance. Okay, equipment maintenance within the aseptic constraints. Next slide quickly. Maintenance in aseptic area must be balanced. Reliability needs with contamination control requirements. Okay, we have to always ensure that contamination control requirements are met or not. Preventive maintenance is always preferable to emergency repair during production. Always be a predictive maintenance. Now people are talking about predictive maintenance, not preventive maintenance. So be proactive. When maintenance is necessary, tools, parts, personnel, and intervention itself must be controlled and documented. When there is, I'm not saying that maintenance is not always required. When there is a maintenance required, but you need to ensure proper process, proper tools, proper person who is qualified. All this needs to be properly documented also. Cutting corners in maintenance can create contamination events that are even more serious than normal operational deviation, guys. So do not do any shortcuts in the maintenance activities. Okay. Emergency procedure and contamination response. I give lot of information in the slides purposefully. There is no visuals. So I'm just reading the important points. I'm just explaining the important points. So don't need to go through everything. Um, but those are a very good information. When contamination is suspected, the first action must always be to stop production and secure the situation. Okay, first action is stop the production and secure the situation. Next steps are assessment, root cause investigation, corrective actions, and communication. A strong response system prevents a local event from becoming a broader quality failures. That's the point. Okay. A true test quality culture is not whether an event occurs, but how organization responds when it does. Very important is like how we are responding to the emergency issues, emergency contamination issues. Okay, immediate response, stop. Assessment, root cause investigation, implement kappa, communication and follow up. Okay, 15 minutes to initial response, quickly. 24 to 48 hours initial assessment, complete it. Complete your investigation within 30 to 90 days. Okay, that's the point. This is my experience. Okay, I'm not saying anything about the your standard, any standards. You can also reduce it. Okay, quality control sampling in a grade A environment, very important. We don't always take care about the sampling procedures in the grade A environment. So sampling is necessary for verification, but it also creates an intervention risk. That is why sampling plan must be validated, representative, and performed with the same aseptic discipline as production. Okay, please remember this. Sterility sampling, visual checks, and in-process control must always be documented carefully. The goal is to obtain valid information without compromising the integrity of the batch. That's the important point here from this slide. So the same manner, whenever you perform any sampling in the gradient environment, you have to validate those sampling techniques, okay, and those interventions. Still 10 more slides quickly. I will go through and then we will leave it for question and answer session. Okay. Cleaning and disinfection protocols. Again, we touched many things now. Maintenance topic we touched. We talked about the QC sampling. We talked about the operator interventions. Now we are touching cleaning and disinfection protocols. Very, very important, guys. Cleaning, disinfection, and sterilization are related, but they have a distinct activities, and each has a specific role. All are not same. Cleaning removes residues. Disinfection reduces bioburden, and sterilization destroys all forms of microbial life. Okay. Effective programs also require correct agent selection, surface coverage, and enough contact time. All these points you have to remember this whenever you perform cleaning and disinfection. Poor cleaning practice is one of the fastest ways to undermine a validated contamination control system. Okay, very important. Poor cleaning practice will lead to a contamination events. Okay, contact time, agent rotation, proper application, coverage is also important in cleaning and disinfection practice. The role of environmental monitoring data, we all know that we all have a 4 types of sampling: viable, non-viable, settle plates, and etc. Particle monitoring, etc. So what I'm trying to say from this slide, environmental monitoring data transforms invisible contamination risk into measurable information. You get lot of information from this data. It includes viable monitoring, non-viable particles, and key environmental parameters. Okay. The real value comes from interpretation, investigation, and action, not from the data collection alone. You need to properly interpret it. You need to properly investigate it. You need to properly implement your action plans. When all these are done, when teams understand the meaning of the data, they make better behavioral and operational decisions, guys. So you just don't need to trend everything, sign off the record. You need to understand it, interpret it properly, investigate it properly. Then you can able to control the contaminations in the clean room. Okay, now let me move quickly. Trend analysis. We all know this trend analysis and corrective action, but very shortly I will tell what I'm trying to say here. Trend analysis helps identify slow deterioration, okay, before it becomes an excursion or failures. Trend will give us a very meaningful data. It will give us information that something is going to happen wrongly. It requires a data aggregation, statistical analysis, okay, pattern recognition, root cause investigation, and effective kappa finally. When you get all this trend, you can able to do entire process and interpretations properly, then the results are good. Results can still be in specification, okay? Because you always see it is in specification. That's a good case study in my past webinar also I did. We did not reach the alert limit, but if something goes upward trend towards the alert or action limits, so that gives some meaningful data for you. You need to be very careful from that information, even though if it is in specification, okay? Mature sterile facilities act on trends early instead of waiting for a formal out of limit event. So what I'm trying to say, mature sterile facilities always act on trends. They don't wait for the event should occur. That's the key message. So properly trend analysis and corrective action will helpful for controlling the contaminations in a clean room. So there are a lot of regulatory expectations from MHRA, EMA. I will not read again. MHRA, everything is a common message. Proper behavior, proper contamination control strategy, validated media fields, meaningful environmental monitoring. Meaningful environmental monitoring. Please understand my words. Meaningful environmental monitoring. Okay. Inspectors increasingly focus on behavior, investigation, quality of the investigation. Okay, and management alignment with quality commitments. A facility with advanced equipment. This is very important point. Facility with advanced equipment but poor discipline will still attract serious inspection concerns. That is what I'm trying to say from this slide. You have a lot of robust robust equipments, robust designs, everything, but improper or poor discipline in aseptic areas will be a serious concern. FDA guidance on aseptic processing. There is a very good guideline. Everyone must read, and it's still current. 2004 is still current. We all know that. FDA guidance is emphasis that sterility must be built into the process, not tested into the finished product. This is why media fields, environmental monitoring, facility designs, training, and investigation systems are so important. FDA inspection often include prolonged observation of actual operations and operator behavior. Okay, so agency expects not only compliance. Key point again, agency expects not only compliance but also scientific justification and consistent execution, guys. That's very important. That's what this slide particularly says. Okay, there are a lot of information I have mentioned from many of the research articles, but the key message, not only compliance but also scientific justification and consistent execution in aseptic processing. So ICH-Q7 guideline, also very good guideline. You can always read this. It's available in web page. It's free of cost. Poor observation and feedback system, very important, very, very important. Poor observation is a powerful way to strengthen behavior. Sorry, peer observation for the poor aseptic practices. Okay, so peer observation is a powerful way to strengthen behavior because colleagues often notice practical risk first, because your colleagues knows what you're doing, whether you are doing it in right area or wrong. So gowning procedure compliance, hand hygiene, glove port intervention technique, material transfer, response to environmental monitoring, documentation completeness, equipment handling and cleaning practices, general aseptic behavior. So all this peer observation is very important. Okay, observation, immediate feedback, discussion and coaching, documentation and trend analysis. So peer observation works because feedback from colleagues often carries more weight than supervisory direction. Your colleagues can support you. Your peers can tell you what you have done wrong. Then you have to correct it. Peers works alongside each other daily. Understand practical challenges. Okay. When a trusted colleague suggests, I have found these techniques works better, operator listen differently than when supervisor mandate you must do it this way. So please get your feedback from your peers about your aseptic techniques or aseptic practices. Self-assessment tool always helps operator reflect on readiness behavior. Now I'm touching the self-assessment tool. Before it was peer feedback. Now I'm talking about the self-assessment tool and checklist. Pre-shift check, personal readiness, knowledge verification, procedure review. Before your shift starts, do this. Post-shift, what went well, what could improve, any near miss or lessons. You always need to discuss about your post-shift also. What went well, what could improve, any near misses or lessons. And during the operation, periodic technique, self checks, environmental awareness, documentation, current documentation. Weekly review, aggregate a self assessment data, identify patterns and self improvement goals. These are all things you can do by yourself. You can have a screenshot of this slide. It's very interesting one. For example, I will read only one point. Personal readiness. Am I well rested and alert for today's operation? You can always do your own self assessment checklist. Are my hands in a good condition? No cuts, no issues, excessive dryness? Am I free from illness that might compromise performance or contaminate product? You have to ask these questions to yourself when you are working in a aseptic area. Do I understand today's operation and any special requirements? Have I reviewed recent deviations or lessons learned? All these things you have to ask by personal readiness. Knowledge verification. Can I explain the purpose of each procedure I will perform? You know this. Do I know the alert and action limits for the critical parameters? Am I clear on how to respond environmental monitoring alerts? Do I know whom to contact if the questions or problems arise? Okay, these are some important daily checklists. Technical self-assessment during operation. You also need to know this. There are some nice questions I have mentioned here. Monthly competency self-evaluation. You also have to go for a monthly self. I'm talking only about your aseptic practices, guys. Okay, all you are doing in the clean rooms. Okay, I will not bore you guys for long presentation. Let me move quickly on the leadership roles topic quickly. Leadership has a powerful effect on the aseptic culture because people watch what leaders reward, tolerate, and ignore. Visible commitment, proper resource allocation, and consistent accountability all shape everyday behavior. Okay, visible commitment, resource allocation, accountability, and consequence. If leaders prioritize production over quality, operators will feel that pressure immediately. Okay. If leaders consistently support quality decisions, the culture becomes stronger and more trustworthy. Stop production when quality is questionable. Invest in contamination control improvements. Celebrate quality achievements publicly. Participate personally in investigation. Ensure adequate staffing and training. So all these are leadership actions. Okay. Listen to operators' concern and act on them. Never compromise standards for deadline. Very important. Admit mistakes and implement corrections. Okay. This is the key message. What leadership should know this in terms of aseptic processing. Building a culture of accountability. A culture of accountability means
Varad, I'm sorry for interrupting, but we
yeah,
are way rolling over the schedule.
yeah, 2 minutes. I will close it. A culture of accountability means people do right thing because they believe in it, not just because they are observed. Okay. The culture requires leadership support, practical systems, and psychological safety. People must feel able to ask questions, report issues, and speak up without fear from the clean room point of view and for everything. In the best sterile environment, accountability is both personal and collective. Okay. This is the most important slide. Please raise your concerns if you see any potential quality issues. Ask questions, self-reflections. All these are important points. That's what I tried to explain. But I will skip this slide. Real world scenario. This is very good scenarios I mentioned here from my experience. Cloud on, particle alert, shortcut requests, uncertain procedures, and peer pressure. So I mentioned here situation. What is the temptation thing? What is the right decision? This concept, this is what I tried to explain it here in a very nice real world scenarios. Okay. Key takeaways, last slides, Igor. I will complete this slide quickly. The key takeaways, aseptic behavior is a mindset, not just a procedure. Okay. To be memorized. The isolator does not replace technique. Environmental monitoring must be taken seriously, and media film must be used as a learning tools. What people do when nobody's watching reveals the real quality culture, guys. Continuous observed scenario based learning is the best defense against contamination. Continuous scenario based observed training is the foundation of contamination free manufacturing, which is very important one. Okay, from my side, sterility is not achieved in the batch record. It is achieved in the mind of every person who touches the process and sustained by culture that surrounds them. This is my statement. It's not achieved by batch records. Okay, thank you so much for attending my session. And there is a book, my book. You can always use this link, and you can also connect with me in LinkedIn. I hope this session has strengthened not only your technical understanding, but also your behavioral awareness. If today's session is like, helps even one person make a better decision in the critical moment, then I have done it well. Thank you again, and I wish you continued success in protecting product quality and patient safety. That's it, Igor. Thanks, everyone.
Thank you so much, Varad, for your presentation. Let's go through one questions, but very, very briefly. If you can stop sharing and go to the Q&A box, uh, read it for us and address it, please.
I will just quickly see the quick Q&A box. Okay, there is one question. What is the importance of PubSit requirements in Europe regulatory? It is not feasible due to facility. That's totally fine, totally fine. But in one of my interaction with EU inspector, see, if you see Annex 1 also, it doesn't say that you need to do uh, okay, ask you to start your video. Okay, let me show my video then. Sorry. Okay, you can see my video. Okay, to answer this question quickly, of course, you can definitely do your risk assessment why you cannot perform PubSit. Okay, but it should be scientifically sound, and you need to try everything that it's leading to a contamination. If you do PubSit in your because of your design issues that may lead to contamination risk, then you can definitely prove, and with your proper risk assessment tool, you can justify that you are not going to perform PubSit. But Europe regulatory first requirement is to perform PubSit. Is again, it is not mandatory as per Annex 1, but if you're not performing, you need to justify in detail. Okay.
Mhm.